my name's Ross Graham. I'm a senior lecturer in genetics at Curtin University in Western Australia. When I'm not lecturing in genetics, I'm a medical researcher and I research into liver disease, and in particular, I'm looking at the role of liver disease in obesity. Now, just because this is medical research and because this will end up on the Society's YouTube channel, I just want to put the following disclaimer in. I'm not a medical doctor, and so nothing in the following presentation constitutes medical advice. So obesity, we've all heard about obesity. And this is data from the Western Australian Health Department, which shows that over the last roughly 20 years, the rates of obesity in WA have been increasing over time. Now, this isn't unique to Western Australia. If you look at data from other states or from other countries around the world, we see a similar pattern occurring. There's also a fairly steady number of children who are classified as obese. And this is not only a problem for the individuals concerned, but it also becomes a problem for the health system because it costs money, and it's also a problem for the wider economy. Because these people, people who are obese are at higher risk of other diseases like heart disease or stroke or diabetes or various forms of cancer. So obesity is a disease of multiple organs. This picture has been taken from a review talking about clinical problems, but those clinical problems can be, I suppose, associated with organs. So for example, diabetes can be associated with the pancreas, hypertension with the cardiovascular system. There's also the reproductive system, the heart, and in particular, what I'm going to be talking about today, which is the liver. So fatty liver disease is essentially the hepatic manifestation of obesity. We're very original in naming these things. And I'm particularly interested in non-alcoholic fatty liver disease. So that's any form of fatty liver disease that's not caused by the excess consumption of alcohol. So you'll hear me referring to this as NAFLD, and it's a collective term for a set or a spectrum rather of chronic liver disorders. And this picture here just shows how fatty liver disease can progress. So a healthy liver can accumulate fat. Once you get more than about 5% of fat in hepatocytes, that's classed as steatosis. And for those of you who like looking at the histology, this is an H&E stain section. We can see these white dots here, which represents the fat in the liver. Now that in itself isn't too much of a problem. The liver can store fat, that's not a problem. It's when that fat becomes damaged and causes damage to the cell and damage to the tissue surrounding it. And this progresses then to steatohepatitis. And that's essentially when the liver starts laying down scar tissue. So this is collagen. And this is reversible. So if the patient takes the advice of their doctor, then this can be reversed. But quite often it just continues to progress, and once this collagen starts to join up, it can then form a ring around the functional units of the liver, and this is called cirrhosis. Now, what this diagram doesn't show is that about 2% of people can also progress to hepatocellular carcinoma, which is liver cancer, which is obviously very serious. Now, I'd just like to take a little bit of a detour, but it's a relevant detour because what my lab does is we look at the association of iron with other diseases in the liver, and in particular, in this case, fatty liver. So iron and fatty liver are associated with one another, and I'll come to that in a minute. But iron is important because it's essential for life. There's no life on Earth that can't do without iron. It's particularly important for things like DNA synthesis, which is why I like showing this picture. So this is a picture of the DNA tower up in a place called Kings Park in Perth. If you've been to Perth, you may have climbed to the top of the tower. The views are great. So once the border opens, I recommend if you are coming to Perth that you climb to the top of the tower and look at the view. But the reason I like to show this is because it's made from steel, and steel is a form of iron. So this engineer's version of DNA is made from the material that is so important for the biological version, because iron is an important component, is an important cofactor in many of the enzymes that are used to catalyze either the recycling or the biosynthesis of DNA. DNA synthesis is not the only thing that iron is important for. It is important for things like nitrogen fixation. So nitrogen fixing bacteria will use iron in their nitrogen fixation pathways. It's also important for things like photosynthesis. So the oxygen we breathe is associated with iron. If we breathe in that oxygen, it's transported through our bodies, associated or rather bound to red blood cells, and it's bound to the iron in those red blood cells. It can then be donated, the oxygen can then be donated to tissues, and eventually it can be turned into energy via the electron transport chain, and that involves iron. So iron is essential for many, many components of life. So I suppose a slightly more sober way of saying that is that iron is an essential trace element. Iron is stored in the liver. So as it's taken up by the gut, the first organ it hits is the liver. It's taken up by the liver, it's stored there, and it's then distributed throughout the body. And the liver is a very important organ in the regulation of iron homeostasis. But the liver is also the site of many other metabolic processes, and it's very likely that these processes can interact in some way, and sometimes they can clash. So what my lab does is that we investigate the interaction of iron with other metabolic processes, and that includes non-alcoholic fatty liver disease. So I mentioned a direct link between iron and fatty liver disease. If I go and talk to my clinical colleagues, about a third of the patients that turn up in their clinics with NAFLD have altered iron parameters, and that includes increased iron in their livers. We don't know why, we don't know the mechanism, it just is, it's a fact, it's there. But the iron that's there is often associated with the progression of NAFLD. So going from this steatosis state where there's just fat in the liver to damage to the liver caused by, or damage to the liver resulting in steatohepatitis. So the damage is often caused by the fact that iron can redox cycle. So it can donate electrons and it can accept electrons. Now this is a normal part of its function. All of those functions that I showed, before involve redox or donation of and acceptance of electrons by iron. However, if that electron is donated in an uncontrolled manner, it can react with other molecules, including the lipids in the liver, and it can damage those lipids. If you get too much damage, it can then damage the cells, and that can damage the surrounding tissue. And this is considered to be one of the mechanisms of progression to steatohepatitis. However, over the last few years, we've been postulating a role for iron in the initial lipid accumulation. And so in other words, in this going from a healthy liver to a steatotic liver. And so I'm going to talk about the initial finding for that, and then give a bit of brief information about how we've accumulated that evidence. So this came about when I had been doing some, when I'd been experimentally loading livers with iron. So this is a graph showing the amount of iron in those livers. This in blue is normal liver, and in yellow is iron-loaded liver. There's about a fourfold more iron in the iron-loaded livers than there are in the normal livers. The pink here is iron-deficient liver. I'm not going to be talking about that today. I'd also like to draw your attention to this. This is ferritin. Ferritin is the iron storage molecule, for those of you who may not know. It's essentially like a soccer ball. It's a cage. And so the more iron you have, the more of these cages you need to store it. So a single one of these ferritin molecules can store up to about 4,500 atoms of iron. And if we look at the Western blot here, this is the amount of ferritin we're seeing in the normal liver, but there's a lot more in the loaded liver. And that shows that this iron loading, this experimental iron loading of these livers is acting in the way in a way we would expect. So what I did was I took I did a gene expression analysis on this using what at the time was cutting edge technology of microarray. Nowadays you do this by RNA-seq, but at that time microarray was the thing we used. And so we got the data back from this and we analyzed it. One of the ways we analyzed it was using gene set enrichment analysis, or GSEA. And what this does is it doesn't just look for individual genes that are upregulated or downregulated. It looks for clusters of genes, so genes that can be that form for example, form a pathway. And so say 70% of genes in a particular pathway may be upregulated, then that gives that pathway a high score in terms of the entire pathway being upregulated. And so when I got the data back for that, what was sitting right at the top, but biosynthesis of steroids. And a bit further down was cholesterol biosynthesis. So there's quite a bit of redundancy in the way GSEA looks at its, or tells you about these, lists its pathways, I suppose. But that gave me a lot more confidence that this was a real finding. So that was the first bit of evidence that I had that iron was associated with lipid accumulation in the liver. So I had to go and confirm that, which I did by looking at gene expression of the cholesterol synthesis pathway. Now, this pathway contains about 24 enzymes. I'm not going to go through them all because it would get very boring. But what I will do is show you a couple of representative examples. So if we look at the gene expression of HMG-CoA reductase, which is one of the main regulatory steps in cholesterol synthesis, gene expression increases with iron loading. If we look at the next enzyme in the pathway, PMBK or phosphomevalonate kinase, that also increases significantly with iron loading. Of the 16 genes that I measured, about half of them were upregulated, and that gave me good confidence that the pathway was also upregulated. Apart from these 7, there are another couple that just failed to hit statistical significance, but in the context of these results, they were probably biologically significant. Okay, so I'm a biochemist, but I'm also a physiologist, and the physiologist part of me wants to see function. So it's all very well for the pathway to be upregulated, but does that result in more cholesterol in the liver? So I went and measured cholesterol in the liver, and yes, in fact, it did. So total liver cholesterol increased with increasing iron. And so I had evidence that said that iron loading causes an increase in cholesterol biosynthetic enzyme gene expression as well. And this is functional. So there's an increase in liver cholesterol. And all of this was consistent with the role for iron in the initial lipid accumulation. Now, since that time, we've also accumulated more evidence, particularly surrounding things like triglycerides and fatty acids. Now, I'm not going to show you that today because the title of my talk was One Cell at a Time. What we wanted to do, because NAFLD is essentially a cellular disease, these processes are happening at a cellular level. And so I wanted to know, could we look at a cell model and could we see changes in lipid accumulation? So we wanted to set this model up to ask questions. First of all, can we see changes in lipid metabolism? If so, how fast do they occur? And thirdly, can we see these changes in real time? So the model we used was a fairly common model, AML12 cells. They're an hepatocyte cell line. I'm not sure if you can see them very well, but this is a bright field image of some of those cells. And for those of you who do tissue culture, we can grow them in the normal way that you would grow cells in a Petri dish. I'd just like to point out this green square here, or in fact, not the green part, but this bronze part in the middle. This is silicon nitride. And so we needed, for some of our downstream experiments, we needed to know that these cells could grow on silicon nitride. And I'll talk about that in a minute. So we could add soluble iron, or obviously the control vehicle, or any other reagent that we wanted to these cells. We can then incubate them, and then after the appropriate amount of time, we take them out of the incubator and perform our imaging or our experiments on them. So the things we wanted to measure, first of all, were iron itself, but also obviously lipids. We wanted to see if there was any accumulation of lipids. So we're lucky enough to have the Australian synchrotron facility over in Melbourne. And we were able to apply for time at the Australian synchrotron to conduct these experiments, and we've been very successful doing that. So to measure iron, we can use X-ray fluorescence. So XFM, the XFM beamline, X-ray fluorescence microscopy, and this was the reason for making sure that cells could grow on silicon nitride, because silicon nitride is invisible to these X-rays. As it happened, they do grow well on silicon nitride if you don't pop the silicon nitride membranes first, because they're very delicate, but they will grow on that. And we were able to then image the cells. Now I'll just point out the color scale here. More red, more yellow means that there is more of that element. So we can use XFM to look for these elements. So if you look at the bottom panel, this is chlorine. The reason I've shown this is because chloride is ubiquitous in cells, and so it gives a nice outline of the cell. So this is a single cell. But we're interested in iron. So if we image iron from the same cell, after 12 hours, we see this amount of iron in the iron-loaded cells, and this amount in the control cells. If we look at our 24-hour cells, we see that iron loading has been maintained. And this is quantitative. And the quantitative data backs up the data that we get from these representative images. Iron has loaded by 12 hours and it's maintained at 24 hours. And so that was good. That meant that for the time period that we were looking at, the time period we want to measure lipids, then we didn't have any problem with our iron loading. The model was working as we were expecting it to. I'll also point this out at the bottom. The intracellular iron showed a punctate distribution. Now, the resolution of the physical resolution of these images isn't enough to be able to see the ferritin molecule, obviously, but ferritin molecules tend to cluster. And so the punctate distribution is consistent with iron being stored in ferritin. So that gave us a little bit of extra confidence to say that the cell was taking up the iron and it was doing what a cell would normally do with iron when it stored it. And so we can have more confidence in the results, knowing that the cells are doing what they should. So the thing we wanted to do was to measure lipids. We can use infrared radiation to measure lipids. And we did this at the infrared beamline, which is about 50 meters around the ring from the XFM beamline at the Australian synchrotron. So the beamline scientists there, I believe this was driven by Mark Tobin, have a lovely setup where they use a modified flow cell to grow live cells and then image those live cells with infrared radiation. And so we were able to use this system to grow our live cells, our hepatocytes, and try to image lipids. So this is the setup. This is the microscope here. This disembodied hand is my PhD student keeping the detector cool. But the important thing here is this perspex box. So this is the stage. This is the microscope stage. If we zoom in on that, this is the objective lens, and this is the flow cell that I was talking about. You may just be able to see some pink coloration in that tubing. That's the experimental medium in the in this flow cell. So if we look at an exploded image of the flow cell, the important component is this thing here, this disc. So this is a calcium fluoride disc. So one of the things we needed to work out was whether the cells would grow on calcium fluoride discs in the same way that we found out that they could grow on silicon nitride wafers. And in fact, yes, they do. They grow quite well on these discs, which means that we can take our live cells over to the synchrotron, we can grow them up, we can plate them down onto these on these calcium fluoride discs. And after we've started our incubation at the appropriate time, we can take those calcium fluoride discs out, we can seal them in this flow cell, and then we can conduct infrared measurements looking at the changes in lipids. So if we look down the microscope, this is what we see. We see a bunch of cells in bright field, and in this case, we've chosen to image this cell. So we're imaging a single cell each time. So just a little bit more background before I get to the results. Every time we take an image, we end up with a spectrum. So this is spectroscopy, and these and the spectrum and the peaks in the spectrum show us are important. Sorry, getting tongue-tied. My apologies. The peaks in the spectrum allow us to quantitate different bonds in biological molecules. So, for example, up here I've shown a fatty acid. This is palmitoleic acid. We can measure the carbonyl ester at this point in the spectrum. We can measure the CH2s at this point in the spectrum. We can measure olefins, which is double and triple bonds, but in a biological context, that's going to be almost all double bonds, and so we interpret it as double bonds. And we can also measure the CH3 group at the end. We can drill down into that a little bit further as well. I'll talk about that a little bit later, but for the moment, these are the important bonds that we want to look at. So when we do that, what do we see? Well, this is a bright field image of a cell. This is the methylene map, so the CH2 map of the same cell. I'd like you to keep an eye on this brown spot here. That's a lipid droplet, and the equivalent is this red spot here in the methylene map. That's this same lipid droplet. And I'll also just point out the color scale down here. Anything more yellow, more red means more lipid. So when we track this over 8 hours, this is what we see. So immediately, if you have a look at this brown spot, it appears to, the lipid droplet appears to separate. Half of it disappears up this process, this cellular process, and the rest of it sort of remains where it is, or maybe heads back down into the cell body here. We can see the same thing on the methylene map. The lipid droplet seems to split. Some of it goes off up this cellular process. And this, we were very, very happy when we saw this, because people are so used to seeing static images of cells, they forget that you tend to forget that the cell is a dynamic entity. It's moving, and the things inside it are being moved around. And so we were able to see this movement. We were able to track the lipid droplets. And as I said, we were very, very happy when we saw that. Now, this is quantitative. We can, or at least semi-quantitative, we can measure the amount of lipid that we're seeing here. And so if we do this for triglycerides, then the first thing we notice at 16 hours, which is the first time point that we imaged, is that the iron-loaded cells had about 3 times more triglyceride in them than the control cells. And this was maintained over 8 hours. And in fact, the amount of the amount of triglyceride increased over the 8-hour time period that we looked at these cells. So we were seeing more triglycerides in iron-loaded cells, and those triglycerides accumulated over time, whether there was iron loading present or not. And that makes sense because the control cells will certainly be taking in nutrients, and they'll need to do something with it, and that will include producing things like triglycerides. If we look at some of the other bonds, if we look at the CH2 and the CH3 bonds, we see the same thing. So we're seeing more in the iron-loaded cells than in the control cells, and these tend to continue to increase over time. So we can look at this data a little bit more closely. So most fatty acids will have a single CH3 group at the end. They will also have a variable number of CH2 groups in the middle. The more CH2 groups they have, the longer they are. And so we can get an estimate of the relative chain length if we take the ratio between the CH2 groups and the CH3 groups. So if we do that, this is what we see. In the control cells, the fatty acids seem to be getting shorter over time. There's a decrease in the chain length, the relative chain length. In the iron-loaded cells, the chain length appears to be increasing. So not only is iron causing an increase in the amount of fat, it's actually causing those fats to become longer. So it's another way of storing the carbon units. There are longer chain fatty acids in iron-loaded cells than in controls. Now, I also mentioned we could drill down a little bit further into the spectra. And so one of the things we can do is look at double bonds. If we take the second derivative of the spectrum, there is a trough around the sort of the 3010, 3015 point. In the control cells, this trough is at 3011 wave numbers. In the iron-loaded cells, it's shifted to about 3014 wave numbers. This indicates, this trough is indicative of the number of double bonds per molecule. What this indicates is that the iron-loaded cells, the fat in the iron-loaded cells is becoming more polyunsaturated. So there's more double bonds being introduced into the fatty acid chains than there are in the controls. And so in iron-loaded cells, fatty acids are more unsaturated. Now this may make those cells more susceptible to oxidative damage. So one of the things I didn't say earlier on when I was talking about how oxidative damage occurred is that the substrate for the electron, so the thing the electron will target, is the double bonds. So the more double bonds, the more likely it is that an oxidatively damaging reaction is going to occur. So it appears that iron is increasing the length of the double bonds. It's increasing the length of the fatty acid chains, and it's making them more susceptible to damage that it itself can cause. Okay, so in conclusion, we've successfully imaged live mammalian cells over a period of 8 hours at high resolution using infrared microscopy. We can distinguish between differences between lipid types and also changes in those lipid types. And using this system, we've shown that iron induces lipid accumulation within the first 16 hours of incubation. And we've also got some information that I haven't shown today that indicates that it's probably somewhere between 8 hours and 16 hours of incubation. The lipid concentration increases over time with or without iron loading, but there are longer chain fatty acids in iron-loaded cells than in control cells. And those longer chain fatty acids in the iron-loaded cells appear to be more unsaturated than those in the control cells. So the control cells seem to have shorter fatty acids that have fewer double bonds, whereas the iron-loaded cells have longer chains that are more polyunsaturated. So in conclusion then, our single cell model has provided us with some direct evidence that iron loading may be driving de novo lipid synthesis. There is increased fatty acid bond unsaturation in the presence of increased iron, and that may make the cells more susceptible to oxidative damage. Now, I think I've said this, I think it's the third time I've said this in about 3 slides, but it's a very important point, given that the progression of NAFLD is considered to be linked to the oxidative capabilities of iron. And all of this is supporting our hypothesis that it's consistent with the role for iron in the lipid accumulation that eventually leads to hepatic steatosis. So finally then, there are some acknowledgements that I would like to make. The initial cholesterol work was done with colleagues of mine in WA and as well as over in New South Wales. The more recent work on the single cell model, I've done at Curtin University in collaborating with my colleagues here at Curtin. I'd like to acknowledge the beamline scientists at the Australian synchrotron, the IR beamline scientists and the XFM beamline scientists. Without these guys, we wouldn't be able to do what we're doing. They're just amazing people. I'd also just like to take the opportunity, because I've got the time to do it, to also acknowledge the, I suppose, the support staff at the Australian synchrotron. They're the people who are there at 3 o'clock in the morning when you've got a question that desperately needs answering and you can't go on without answering the question. They don't get a lot of acknowledgement, and I just would like to take the opportunity to do that as well. Finally, I'd like to acknowledge my lab members. I've got a great lab group. We make a very good lab team. A couple of these guys have moved on, but I've presented some of their work today, so they're here. And the single cell imaging work that I showed was done by Clinton, who is my PhD student and who is just in the final throes of writing up at the moment. Finally, I'd like to thank funding organisations. The initial cholesterol work was funded by the what is now the Spinnaker Health Research Foundation. Obviously, I'd like to thank ANSTO and the Australian Synchrotron for the beam time that we've been awarded. And also we've obtained some funding from, some internal funding from Curtin University and Sherry, the Curtin Health Innovation Research Institute. So thank you very much for listening.